first session will be about meaning of life, perceiving of life from view of biologists, molecular biologists. So I think you can start.
So hello, what I want to do here today is partly scientific and partly philosophical, and it should be some kind of refreshment for you maybe. For the scientific part, I sure do not want to overwhelm you with quantum of scientific data, facts and terms. For the philosophical part, I do not want to bore you with anticipations I did while I was drunk, although believe me that I have many. But instead, I would like to tell you something about how scientific thinking can sometimes contribute to solving of most puzzling scientific and philosophical questions. I would like to tell you something about life from a view of molecular biology. Molecular biology is the discipline that deals with the molecular basis of biological activity. And in the next few minutes, I would like to explain to you why I believe that molecular level is so important when talking about life. So, this is my name. This is the old Egyptian symbol of life, so-called the key of life, and this is the central dogma of molecular biology, a simple scheme which shows how the information is flowing from a molecule to molecule in all living systems. In my presentation, I would like to explain to you the central dogma of molecular biology, and I will also like to tell you why I personally believe that it could be as well called the central dogma of life. So, speaking of life, we might first want to ask a very important, very fundamental, and also pretty cliche question. And the question is, what is life? When we try to answer this question, our success is almost always relative and almost never absolute. And this is pretty strange because when by just our senses we have no problem to distinguish a living organism from a non-living thing. But when we try to formulate a proper definition of life, such a definition that would cover up all the different forms of life and none of the false positives, we might find it very difficult. And especially when we are thinking on a macroscopic level. To try to define life on a macroscopic level is not only very difficult, but it's also very inappropriate. And the reason is simple. If you ask a random person what are the different forms of life, the answer you will most probably get will be that there are animals and there are plants. This is an understandable answer because animals and plants are sure living things, and the difference between them is obvious. The only problem is that we've just ignored more than 90% of the biomass. This part is presented by unicellular organisms. To be an unicellular organism means that all the life important processes are held by just one cell, and this cell also represents all organisms' body. This is unlike the animals and plants, which bodies consist of millions or even billions of cells. So, to define life, we must go down to the cellular level. At this level, we can describe the cells, we can name them and categorize them, we can put them into different groups. At the present, most scientists believe that every single organism living on this planet can be put into one of the three major groups. They call the groups divisions. The divisions are called prokarya, eukarya, and archaea. Here I choose one representative for each of these divisions. This is Salmonella diffinurium, prokaryote or bacteria causing salmonella. The second is Saccharomyces cerevisiae, budding yeast, which is used in the food industry in making of bread or brewing of beer. And the third is pretty exotic. It's an archaeon. It's a Pieropus fumari. It lives in deep seas by the hydrothermal vents where the temperature is over 100 degrees Celsius. The fact is that all the animals, as well as all the plants and all the multicellular forms of life, belong to the same group as eukaryotic organisms. As Sakharov necessarily, they are all eukaryotic organisms. And the fact also is that the differences between prokarya, eukarya, and archaea are much bigger than the difference between animals and plants. And the reason is simple. This is the evolutionary tree which shows the lines leading to different divisions. And the lines leading to eubacteria, eukarya, and an archaea diverged much earlier than lines leading to animals and plants. And for this long time they all accumulated lots of changes, and these changes are causing the differences between organisms. But one might ask where these differences are, because when we are looking at the cells, we can't see no big difference. To answer this question, we should go one step down to this subcellular level. This is microscopic photographs of anatomy of typical prokaryotic and typical eukaryotic cells. The biggest difference is in organization, internal organization of the cells. While in prokaryotic cell, we can't talk about any special organization at all. All the cell is just one cell space, and all the chemical processes takes place in this homogeneous cell space. Eukaryotic cells are, however, pretty well organized. They have different organelles which are separated by lipid membranes, and specific chemical reactions take place in a specific organelles, like for example, respiration take place in a mitochondria, or photosynthesis take place in a chloroplast. Scientists now believe that...
Excuse me, has this eukarya developed out of the first one? I mean, this is an evolution, the more complex one is the evolution of the first one, or?
I'm just going to say that.
Okay.
Scientists now believe that the internal organization and all the organelles are a consequence of the process of so-called endosymbiosis. It's the process in which prokaryotes were living in a symbiosis, and the symbiosis was so strong that they literally become the part of each other. And so all the organelles are just prokaryotic cells, and they are in there, and they are stayed there, and they will stay there forever probably. But when we are talking about life and about life essentials, maybe more than to be looking for the differences, maybe more important is to find the similarities between these cells. Find such a similarity that all the cells, all the living cells have in common. And one similarity, maybe the greatest impact, is that all the cells store their genetic information in the same linear chemical code, the molecule of the DNA. These two guys are Watson and Crick, which discovered the structure of DNA in 1953, and in my opinion, it's the most significant scientific discovery of at least last 100 years. There is much controversy about this discovery. Some people believe it's all based on stolen data, and some people say that they made the discovery while they were on LSD. But one way or another, knowing the structure of DNA started a very fast development of molecular biology, a young scientific discipline that already improved the quality of a human life significantly. Watson and Crick, however, didn't find the DNA. The DNA was known a long time ago since the 20th century, and it was also known that it's a polymeric molecule consisting of 4 monomers: thymine, adenine, guanine, and cytosine, or just T, A, G, and C. But nobody at that time could imagine how so simple molecule can be essential in the process of transfer of genetic information. A few years later, it was proven that it really is the DNA, the molecule responsible for the transfer of the genetic information. But how is this possible still remained a mystery. When Francis Crick and James Watson discovered the structure of the DNA, everything seems much clearer. DNA is a linear double helical molecule, and nucleotides are arranged in a specific order there, and the order somehow codes for the genetic information. The DNA is a double-stranded, and the strands are complementary. Complementarity means that each strand is a negative to each other, and this is due to the special chemical properties of the nucleotides, which guarantee that T is always paired with A, as G is always paired with C. And this is very important for the process of DNA replication. All cells replicate their genetic information by completed polymerization. In this process, the DNA is depleted, and to each strand, its negative is synthesized by the laws of complementarity. So at the end of this process, this one-step process, we have two identical molecules of DNA, and this is important in the process of cell division, when the genetic information has to be distributed equally. The equal distribution, however, is not 100%. During the polymerases of DNA, the DNA polymerase makes random errors, and these errors are as well as errors in the distribution of genetic information during the cell division, cause the mutations, which are the main source of variability in population. And this variability is essential when a population should undergo the process of evolution, which is a basic property of life as well. Now we know how the genetic information is stored and how it is replicated. So maybe we should say what kind of information is this and how it is realized. The realization of the genetic information happens in two steps, two subsequent steps called transcription and translation. In first step, a short segment of DNA coding for a specific product is transcribed into molecule of RNA in a process very similar to DNA replication. RNA is as well complementary to one of the DNA strands, because only one strand of the DNA has sense. So the molecule of RNA is a single strand. Molecule of RNA is very similar to molecule of DNA, but it is much more dynamic. And because of this, genetic information can be stored or preserved in a molecule of DNA, and at the same time it can be realized from the molecule of RNA. We should maybe say that not all the regions or not all the sequences of DNA are transcribed with the same frequency, just as some segments are transcribed almost constantly. The other segments are not transcribed at all because they do not code for any information. What is on the right is the picture of the DNA that is just being transcribed. The axis, or how to call it, is the molecule of DNA, and the outgoing fibers are the molecules of RNA that are just being synthesized. So when the RNA is completed, it should undergo the process of translation. The mechanism of this process is a bit more complicated, so I decided not to show it at all. But what is happening is that complex of many proteins and many more molecules with a complicated structure is literally reading the molecule of RNA, and for each combination of the three nucleotides, a specific amino acid is added into the growing polypeptide chain. The polypeptide chain can then form a very various structure in space, and they can create complexes with each other and create the proteins. So we can say that proteins are long chains of amino acids. In all the proteins that can be found in living organisms, all the proteins consist of 20 amino acids, and they have different chemical structure. And as a consequence of this fact, proteins, unlike the DNA, can have very different structure and very different shape. And it is the structure and the shape which is important for the proteins. Different proteins can play a different role in different cells. This is, for example, an insulin. It's a hormone. On the right, there's hemoglobin, which acts as a molecule for transport of oxygen. But the most unique feature of the proteins is their ability to catalyze specific chemical reactions that would never take place without the presence of the protein. One of such chemical reactions is the DNA polymerases, which we already mentioned. So as the DNA polymerase is essential for the process of DNA replication, thus many more proteins are essential in the processes of transcription and translation. So here we come to an interesting conclusion that the part of the genetic information codes for instructions that instruct the cell to replicate and realize the information itself. In other words, the DNA codes for the proteins that are essential for transferring the codes into the proteins. And this is, in my opinion, a very important fact and very important feature of every single living organism and of life itself. By the modern methods, we are able to read the genome sequences of different organisms, and by the computational methods, we can look for the segments that codes for proteins. And this chart shows different numbers of genes in different organisms. And from this chart, we can make some interesting conclusions or statements. The first one is that the number of genes does not correlate with the overall complexity of an organism. Xenopithecus elegans is a simple worm, and it has about the same number of genes as the humans do. Now, important fact is that the fact that the human cells or human DNA contains about 20,000 genes does not mean that the human body is made from 20,000 proteins, because proteins can interact with each other, they can modify each other, and they can form different complexes. So, as a consequence of this fact, the number of proteins that can be found in a human body is something more than 100,000. And no, not all these proteins are present in all the cells. Protein expression is a strictly regulated process, and as a consequence of this regulation, different cells can express different proteins, and they can have different shape, and thus they can have different function. But we now should take a look at the smallest known genome. It's the genome of Mycoplasma genitalium, which can live with just 500, something more than 500 genes. And scientists think that living cell can exist with fewer than 500 genes. And this was to some part proved last year by Fragment, who constructed the first artificial chromosome. What he did was that nucleotide by nucleotide, he synthesized the artificial molecule of DNA with sequence similar to the sequence of Mycoplasma genitalium. And when done, he transplanted the chromosome into a cell with its own chromosome explanted. And after this process, the cell began to self-replicate and self-propagate. So this very important experiment that showed that it really is the information stored in DNA that is essential for a living. These are 500 genes that are essential for a living. But the question is if really all these genes are essential, and if we
Sorry, when you change the gene, the DNA in a cell that this guy did, do you have to change how many DNA sequences you have to change? Is it enough? Is there only one in one cell?
He took all the DNA and he replaced the total DNA present in cell. I'm not sure if I understand the question right.
There is this sequence of information, and how many times does it exist within one cell, within this most simple cell?
How many times does it work?
Is there only one DNA in one cell, or are there several?
Yes, there is in a bacteria as well as in simple prokaryotic cells. There is one copy of one molecule of the DNA, and also they can have so-called plasmids. It's shorter sequences of DNA, shorter molecules, but they are not essential for life. They can give them some advantage.
Thank you.
So the question now is, if all these 500 genes are essential, and if we can say that these are 500 genes that are essential for living, an answer to this question is both yes and no, because unfortunately life isn't all about self-replicating and self-realizing. To do this, cell has to acquire or accumulate an energy or building blocks to realize what you want to realize. And for this, it needs proteins, it needs genes, and the essentiality of these genes is relative to the environment in which the organism is living. Mycoplasma genitalium choose the strategy of living in a very specific environment, and that's why it is called genitalium. And but the different organisms choose different strategies. This is another evolutionary tree of life, we can call it that way, and the branches of this tree are the organisms that have already been sequenced, which gene sequence of the genome we already know. And from similarities between these sequences, we can count the percentage of similarity, and from this data we can construct the evolutionary tree of how it is most probably the organisms evolved. All these organisms have different set of genes, and they have a different life strategies. But what do they have in common is that they all struggle for a living. They all want to self-realize and self-replicate information they are getting. At the present, scientists also think that every single living organism, every organism living on this planet comes from one ancestor called the last common ancestor, and which is here represented by the center of the cycle. And life itself is a cycle, the cycle of self-replicating and self-realizing. And we as the humans capable of rational thinking are trying to understand these processes to the deepest possible detail. But that does not mean that we are not part of this project called life, a project in which different organisms try to find the optimal strategy for living. And I'm not sure if any organism can be successful in this race, but the sure thing is that if the information flow is blocked, if the information stops flowing from molecule to molecule, the life itself stops. And this is why I believe that the central dogma of molecular biology could be as well called the central dogma of life. Thank you very much.
I would like to ask how many information is in DNA. For example, in megabytes, terabytes, or?
Yes, there maybe is an analogy because the DNA consists from a nucleotide, the basis, so we can say that one nucleotide is 1 byte, I don't know, and it varies from organism to organism. In humans, for example, I believe there's something around about 3 billion of nucleotides, but not all these calls for an information. Some sequences appear to have no role, or maybe we just don't know what are their roles.
So, for example, if we create a great simulator where you're trying to modify some of the sequences and trying to find out what it do, tries this kind of thing, or?
I think people are doing many things with all these, but or to write such a program, or how to call it, will be at least very difficult because we would have to understand all the cellular mechanism and all the chemical mechanisms and all the mechanisms 100%, and this is pretty utopic now.
You told us that there are transcription and translation from DNA to RNA to proteins, so are known some of the rules how from the proteins are creating cells which are, for example, trying to find food or energy or something like that.
I understand.
From DNA you get RNA, from them you get proteins, and these proteins are something which define what the cell will do, and are known the rules how from the proteins.
Oh, okay, I think I understand. For some biological activity they are, and for some there are no rules. That is all very complicated thing, and now we are starting at the very basic principles. Molecular biology has something more than 50 years itself, and it has great achievements, but we are still at the level of single protein, not at the level of interaction. If we want to modify an organism, we can transform a DNA sequence into a different organism, and we know that the organism will produce the desired protein, but we are not. I think we are not able to reprogram ourselves as a whole.
We actually are, aren't we? If you look at iPS cells.
iPS?
Yes, induced pluripotent stem cells. So we can take a cell from the skin and use some transcription factors from the cell and make the cell more primitive to be able to use it as a pluripotent stem cell.
Yes, that's true.
So we are actually right now, since a few years, able to take a cell and reprogram it and make it a completely different cell.
Yes, what I maybe wanted to say is that we can do at this point whatever we want. We are limited by our knowledge.
The question I asked you, I'm actually just want what the information mean when you were talking that was, yeah? And you were giving us information.
Yes.
And when actually DNA transfer information to RNA, what does it mean really? What is actually, I mean, what is the coding and decoding in DNA, which is it's like language we are talking here, or is that the programming, or what? I mean, that is, I really just want to know. Maybe I don't know so much about this kind of thing, but it's very intriguing, very exciting for me. What does it mean really, information?
From a DNA, if I want to make an analogy about transcription from DNA to RNA, it's like something, if you are in a library where you have unlimited general or limited knowledge, and you choose a book or a paper which is interesting, and you Xerox it, and you put the book back at its place, and you take your Xerox with you, and you can then do what you want with it. But the information is still stored in the DNA, still in the library, and you have a copy which you can do.
But what's the transfer? Is it electromagnetic? What is actually this material process? Is it just an electronic magnetic actually waves?
It's all chemical reactions.
I have one question. This is all information storage and information transformation, right? And as such, all of these cost energy. How much energy does it cost to sustain such information system? In general, I don't know if there are any measurements for that, and also for a living organism like humans. How much energy do we invest every day for this information process?
Well, I don't know if it's measured, and if it is, I don't know the answer in exact units or something like that. I can.
But on like a copying process of just the DNA, how much energy that costs?
Yes, I really don't know. I think it's possible to measure that, but I do not want to answer, and I don't want to say something.
Isn't it something like 1 ATP per base? Because the DNA synthesis works in a way that the nucleotides are fused to ATP, and then this ATP is transformed to adenosine monophosphate, so it costs 1 ATP, or two phosphates per base at least, and this is a minimum cost.
But I was thinking about the cost in the exact units, joules or like that.
Is there such a field such as bioeconomics or something?
Yes, there is, and there is actually exact currency, and that is the molecule of ATP.
Wow, cool.
There's actually very, very interesting overlaps between certain economic things and biology. So you can apply certain concepts of economy into biology and vice versa.
Yes,
So a lot of reasons why creatures have evolved the way they have, or living organisms have evolved the way they have, is because of trying to be energy efficient and trying to be economically efficient. So everything you have in your body is highly economically efficient in a way.
So we have another question.
I heard a lot of different stuff about it, but maybe you can explain the basics. So there is this information about how the genes follow each other in the human body, and I know that it costs a lot of money to produce this information, but so what are the license issues involved in this research? So is this whole, like what is the publicly available information, what is public domain need, and what is copyrighted, and what is the dynamics of
You mean when human genome is sequenced?
For example?
Well, this is not part of what I'm interested in the molecular biology, but as far as I know, only 3 or 4 people have a completely sequenced genome, and for example, Ozzy Osbourne has, and I think that he doesn't have, and I don't know, maybe this is very important question in human genetics and human molecular biology, and I think it's still in the stage where it is defining.
This is a good thing that you mentioned Ozzy Osbourne.
People who have the genome sequence. How come that some, I don't know, anthropologists or whoever are so convinced that the genetic differences between people is less than 1 or 0.1%? How can they be so convinced?
This is the difference at the level of sequence of DNA. So yes, I know.
I think
I don't know. You have to ask an anthropologist. I'm not interested in human genetics.
I'd like to go back to the question from Oxy guys about the intellectual property protections on this research. This also raises the other question of privacy, because if someone like Ozzy Osbourne has his whole genome sequenced, this has huge privacy implications because, I don't know, but according to this genome, can I, for example, fake his DNA and plant it at a crime scene?
In theory, yes.
Okay, in theory, that means how economically is feasible is that?
It's actually pretty cheap because the way DNA DNA fingerprinting works in crime scenes is you have a set of pieces of DNA that frequently mutate and they're frequently mutated between individuals, and you have a set of I think 32. I don't know. Do you know the exact number?
What?
Of SNPs you check on DNA fingerprinting. So I think it's about 32 of these sites. So if you take out Ozzy Osbourne's 32 and you just clone DNA, or create DNA, which is incredibly cheap, of these 32 sites and plant it on your crime scene, it's gonna show up as his fingerprint.
But I don't think so.
I think it's possible. I read one article that it's possible to completely fake your DNA.
Oh, you have your own sequence.
Not your own, but I'm sure it's pretty cheap and easy to plant a fake one on a crime
I
scene.
think you don't even need the whole sequence like what Pavel did. He sent it to some DNS analyzer and they showed him that he's susceptible to some diseases and then he's resistant to some other things.
But
I think that's enough for fake his DNA.
No, that's something different. On the crime scene, you really look at these highly mutating sites.
Yes, but they have a sample of his whole DNA, so they can.
Yeah, so they
just fingerprint.
They could check of those.
Just fingerprint. So you just fingerprint some concrete SNP. So it's just hashed or something like that. I used a service called 23andme.com. Maybe you know this service. It was probably the first commercial DNA laboratory. I sent my DNA from Salina to California to this company, and in few weeks that I received my login passport and I can log into my web page and see all my physical characteristics, potential diseases, for example, influence of some drugs or some medical, something like that. So I'd like to ask, I know that at this moment there are also some genetic dating services. I mean, just for matching, males and females. For example, one is genepartment.com. I know about this service. So my question is, how reliable is such dating service? If I want to apply my girlfriend using this service, for example.
Can you diagnose if you're a man or a woman?
Something very similar. Scientists were trying to do something very similar. I think it's called the eugenics, and it didn't work.
I'm just interested in some physical attractiveness.
The question is, how do you match, right? First, it's really hard to get physical appearance from your DNA. We're still not there. Yeah. Although there are people doing prenatal testing to see what color the eye of the baby is and shit like this. But we're still not there to really conclude just from your DNA of your physical appearance.
Yeah, for example, they were able to identify my color of my eyes, color of my hair, and also that I have straight hair, and also they're quite big, for example, and a lot of these physical characteristics I can read from.
But is it this you look into on a woman? So do you look at how the hair patterns?
I know that in
I don't.
Internet, yeah.
Sorry, is your question that can DNA determine to whom are you attracted?
Yeah,
I think yes, and I have heard this. I think I'm not an expert. For me, this is hearsay, so I'm not sure whether it's true or not. But what I heard that you can actually sense through your smell
exactly
whether someone is matching to you or not, and the smell is showing you how close he is related to you in terms of DNA. And the farther away it is, the more attractive the smell of the other one is.
Yeah.
So I think distance of DNA might be the selection.
What about your fantasies, or I don't know, imagination, or art and culture, and all this kind of thing, which say about love and the rest, you know? So there is DNA there, and that determines whom I'm attracted to. So what happened to the rest, actually?
Yeah, no, I think biologically, from a diversity point of view, I think this makes the most sense for the whole population. It's not about culture, we are just one big race, okay?
No, I mean,
if anybody's a racist, then you say, I hate dolphins.
No. I mean, we are talking to individuals. So there is just 4 billion actually people on this earth, you know, so there are 4 billion are grown-up people. So, I mean, my DNA actually determines to whom of these 4 billion I'm going to be attracted?
No, it's not about attraction.
I just want to say that in the
talking
nature,
on parallel levels, actually.
Okay.
You're right and I'm right.
In the nature, it's quite normal and natural, or natural that some animals they attract using diversity, for example, and diversity can be, I think, can be measured by DNA probably. Okay, so if there is some correlation between DNA diversity and attractiveness, and I think there is some kind of correlation probably.
There may be, but I think this is all just statistical thing, but because there are many things that play a role in.
I think we are omitting environment and phenotype because that's also very important. For example, animals don't go to theaters and concerts, so they obviously have only 3-4 types of spending time together.
But but for example, your DNA compatibility can be just one attribute in quite complex data service.
Sure. But I think that's the only one part, and there are hundreds of others. So it's not that important from my point.
Maybe we can find the gene for people who like electronic music. Can match for certain things.
I have a question for you. You said about the dating site with DNA. When you send the DNA to them, do you write if you are male or female on the application form, or do they just find it?
I don't know. I want your preference. What's your looking for?
Well, if they are not able to find out this, then it's probably not a good
I don't know. I haven't tried. Maybe you should. Genepartner.com. But for example, when I
Don't tell them anything. Tell them just, here my DNA, find out what my gender and what kind of partners I prefer.
Could I ask one, not a gene partners question, but more maybe informative question? You said that the DNA is from four parts: guanine, adenine, thymine, cytosine. So the whole sequence of DNA is, can I represent it as ternary code?
What we call
Not binary, but
yes.
Ternary is a reason.
sorry,
Forbit. I mean, Forbit quarter.
Yes.
This is a four-bit process.
Well, sure, you can.
and
Not
why
With
these
lots of
four?
redundancy.
Why these four? Substances is was choosing for encoding the DNA. Are they having some special composition or?
Yes.
Why only 4 and why these 4?
It seems that it's a much more optimal idea to use just binary code. So why do you need 4
Yes,
different
that was the
significations?
meaning of question.
Because you know all these people thinking binary code and they don't understand why this is complex.
Well, as you know, maybe it is just kind of accident because we count in a decimal.
Why 4 bits? Why not 70?
We count in decimal because we have 10 fingers. If we had 12, we would count different. And so I think it's maybe just an accident. Another thing is that
God has 4 fingers.
4 fingers!
Well, maybe.
You have forced
Ask.
behind us, actually.
I would like to ask about those
All contents are
unnecessary
not
parts of
actually
DNA.
Something
Are
very
there any
important.
theories why it is there, or if it really is unnecessary? There is a chance that it is for something that we don't know.
Yes, there is chance. At the beginning, the scientists considered the parts, all these unnecessary parts of the junk DNA. And later, they found that many of these parts really do have a very important role. For some parts, we still don't know role, but that does not mean they do not have any.
Okay, because
We just don't know, and
everything in life is optimized. Yeah, so it would be.
No, everything in life is being optimized.
It's being optimized. DNA or human DNA.
Human life or nature?
Was optimized for millions of years. So why would there be something junk?
Okay.
I
Then there's-
heard something about it.
It's just
they
pizza.
say that you have some probability that few parts will mutate. So if you have few parts which do nothing, you have less probability that you will mutate something which do something.
I can tell you an explanation from Richard Dawkins, which is who is probably one of the most famous genetics. And his explanation is that DNA or some long sequences of DNA, they have tendency to replicate as much as possible in the longest configuration. You know, and it's hard to explain very shortly, but all these genomes have internal tendency to replicate themselves in the longest chain, and that's and because you have many long, probably useless chains, it just means that these DNA chains are successful in this replication process. I'm not sure if you understand.
Well, if I understand it correctly, then being successful in replication and being useful is completely different characteristic.
Yeah. Okay. Being successful.
Being
Or-
useful, not successful.
or useful.
Useful and successful in replication is different.
Excuse me, do you mean actually they produce as many actually links as possible, and some of these links in the west, they don't actually are useful, and some other actually come together, like a factory. You have raw material, they produce, some of the actually west, they go actually throw them away, but there's others that just, come together and actually Richard like that?
Richard Dawkins tried to look to DNA like it is some not human, but something intelligent, something intelligent genome, and the DNA is ever or there's tendency to replicate itself in as much as possible non-change configuration, without any changes. And there is such tendency, and there is, of course, DNA has no erroneous or something like that, but I can send you a link to this explanation. It's quite difficult. But anyway, for geeks, maybe you are interested. I did my DNA analysis a few years ago, and now it's possible. I can download it in plain text. So I can just download my DNA fingerprint, which is in plain text and looks very funny. Just number of genome and then CC or CP or AG or something like that, the combination, and it's about 20 megabytes file. So you can download it, and now at this moment there are some third-party open source software you can use for analyzing these dumps. So it's really cool. So you can just dump and save this your DNA file and use some third-party software to analyze it. And
is there some rhythm in the DNA sequences?
think that we can say that yes, there are some repetitions. Repetitions are very frequent in the genome, and I think that there was one man was trying to add a sound to the sequence. He put a different sound to the different sequences, and then he played it, and it sounds pretty cool.
I would like to have an addition to what Wilder said. Actually, one guy got this 20 megabytes of data pushed into GitHub, and one smartest forked it and started to send him patches. The problem is it's not that easy to apply patches to the genome.
One absolutely great advantage of this commercial DNA laboratory like 23andMe, there is a genetic social network for something like Facebook, but so now I'm able to find, for example, my cousins 300 ago or 400 ago, and I mean, or any ancestor 1,000 years ago. So using this social network, I found a lot of potential family around the world, in the US, Hungary, and all these
How did you
countries.
find your ancestors 1,000 years ago? I don't believe they use the same.
Yeah, but this network is really large at this moment. So 10,000.
I don't believe they have genomes from people.
Okay. I mean, they're children, of course.
So
Well,
there are labs, that people actually themselves. What is how expensive are the labs that people actually can work through this kind of study DNAs? Is it possible that people have say, like hackers actually? It's not run by big just companies. How expensive are the labs actually?
It depends on what you want to do. I'm not sure if
Basic research, if someone wants to do the latest research on DNA, so how expensive is the lab actually?
you want to do what?
Latest research.
Yeah.
Research. Even at Slovakia labs, at the Faculty of Natural Sciences, can afford the lab with everything. It's essential for all the research. I don't believe it.
Okay, we have time for the last question.
Is it possible nowadays to manufacture any DNA string that we can imagine? And second question, if it's possible to connect these artificially manufactured cells or any cells with interfaces in computers and somehow interact with these cells?
I don't know.
I have the answer for you. It's exactly the same as if you would generate random strings and try to feed that to Perl. Sometimes it runs, sometimes it doesn't. But most of the time it doesn't.
Although Perl could happen.
But you have more chances that it happens.
Actually, producing DNA sequences at your own will is incredibly cheap in C.
Yes,
So
but
there
it
are
doesn't make
companies
any
you
sense.
can
It's like
simply order. You just give your
This is how Perl
sequence you want to
Linux,
have,
no?
and they ship you back
characters.
your DNA in a small vial. You can go on and play with it.
Well, if you have a screen, for example, 800 to 600 pixels, and you will try to make all combination, you will find there everything which a human every time in history find out, and they will be everything.
Not everything. Some things are too long for
Yes.
I think we should finish.